Okay, so we're going to talk about genetics for our test. So here's just some vocabulary from the first section. And so this says define traits. So traits are going to be the characteristics that are passed down. Characteristics passed down that are expressed. So, if you are tall or short or brown eyes or whatever, those are going to be the traits that have been passed down that you're expressing. Okay, genetics, this is just going to be understanding genes in our DNA and how they give us an appearance. All right. Purebred. This purebred is going to be, you hear like purebred horses or purebred dogs, and this is when you have two dominant alleles. So no matter what, we know that we're going to be getting a, dominant gene. So if I, cross a homozygous dominant with another homozygous dominant, I am for sure that all of my offspring, 100%, are going to have the same traits. And they will all be dominant, and we won't have to worry about any recessive genes coming out. Okay, so cross is just when you take two different alleles and you cross them and you see the probability. So it's to get probability of what your offspring are going to look like. And this could be in plants, this could be in humans. And then law of segregation. Segregation means to separate. And so what this law is talking about is that genes are going to be expressed separately. And this was one of Mendel's laws. That he was able to show us through his experiment. Okay, if Mendel only examined one trait, do you think he would have developed the law of segregation? Okay, so we know that Mendel looked at a lot of different things. He was looking at flower color, and he was also looking at the peas, if they were smooth or wrinkled. He was also looking at the height of the plant, how tall it was. He was looking at many different things, and because he was looking at all of these different characteristics, he was able to see that these genes were expressed separately, that the purple flower could be tall or the purple flower could be short. He was able to see that the purple flower that was short could have smooth peas, or that the purple flower with short could have the wrinkled peas. And so none of these genes were traveling together, and that's how he got this law of segregation. Okay, why is it important that Mendel started with purebred plants? Well, if he would have started with something that may have had a recessive gene, he might not have been able to track exactly where it was coming from the parents. So he was able to take two purple plants, and he knew that they were purebred, and put them with a white plant. And when he did that, he got both two purple plants. And he knew that this one was a purebred. It didn't have a recessive purple or anything, and so when he got these, he knew that it wasn't going to be blending. It was actually that the purple was dominant over the white. But he did know that the white must be recessive, because the next time he crossed these two purple, he saw that one of them actually came out white. And so that must meant that this purple one had a recessive white flower gene. All right, so now this idea of a gene and an allele. So when we think of a gene, that's the segment of the DNA that is in our chromosomes that we pass on from generation to generation. Also, a gene is what's expressed into a protein. And the allele is going to be it's going to be two genes. It's going to be the genotype. And so you have an allele for brown eyes, you have big B, big B. So this is gene number 1, and this is gene number 2. Okay, so you get them one from your mom's side, one from your dad's side. And so if it's something like this, you know that maybe that came from your dad's side. This might be for blue eyes, and you're not expressing blue eyes, but the next generation has the opportunity to get blue eyes, just like this next generation was able to have a white flower. Okay, and so the allele is going to be 2 of these genes, and an allele is going to show if something is homozygous or heterozygous. And this is really important because this is going to tell us what genes are passed down, especially like when we're looking at a pedigree, when we're trying to track something through a family of relatives to see, which ones may have Huntington's disease or some other trait that we're looking at. Okay, so 2 alleles, or 1 allele, 2 genes. All right, so a genome is going to be all of your DNA. Okay, and so we kind of talked about in class, this is like 20,000 to 30,000 different characteristics or traits that are going to be expressed. Okay, so when someone's looking at the human genome, they're really looking at a lot of traits, and we talked about how this is important to understand that you have so many different combinations of these traits. So you might have 20 to 30,000 traits, but the combinations of these are just going to be so much greater, and that's why none of us look exactly the same. All right, what is the difference between a genotype and a phenotype? So genotype is going to be what your genes say, the allele. So this is the brown eyes, okay? The phenotype is going to be how it physically looks. So we know that big B is brown. So that's going to be the phenotype. And then maybe if it's little b, this is the genotype, and the phenotype is going to be blue eyes. Okay, so this is telling us what the gene says. This is telling us what the genes are going to express, or what's it going to look like. All right, what is a dominant gene? What's a recessive gene? So dominant is going to be something that is more prevalent or is going to come through on the gene. So if there is dominance of brown eyes is always dominant over green, which is dominant over blue. Okay, but I think if you have 2 blue recessive genes, it becomes dominant over brown, or it talks about it in our book. I think 2 recessive blue equals dominance over brown. But dominance is going to be when you have something that's heterozygous. Whatever one is that capital letter, the uppercase letter, is going to show dominance. Now, we learned about some of these exceptions that we're going to talk about later when there might not be something that's dominant or recessive. But in this case, this one is going to be dominant, and this is going to be recessive. And so this is recessive is always the trait that's being carried, and dominant's always the one that's being expressed. Okay, so dominant expressed, recessive carried. All right, explain why organisms' genotypes may be homozygous dominant, homozygous recessive, or heterozygous recessive, heterozygous, but never heterozygous recessive. Okay, so homozygous dominant means it's going to be like big H, big H. homozygous recessive means little h, little h. And then heterozygous dominant is going to be big H, little h. Well, because this big H is always going to trump this little one, there never can be a heterozygous recessive, because if it is recessive, which means they're different, I mean heterozygous, hetero means different. And if they're different, it's always, this letter is always going to trump that one, which is going to be dominant. So the only way you can have something that's recessive is with two of the lowercase letters. All right, Punnett square. So we already kind of did one of these, but let's do another one. So we could do, let's do one for tall and short. So big T is going to be dominant, and little t is going to be short. Okay, obviously the combination of these two is going to be dominant. So let's say we have one that is very tall, homozygous dominant, and let's say the other one is heterozygous dominant. Okay, so heterozygous here, homozygous, these are the same. So we're going to have homozygous dominant. Here we're going to be homozygous, oops, I meant to put this as a small t, oh my goodness, sorry. And then this one is going to be small t. So there is a 50-50 chance here that it's going to be tall or short, just because of this recessive gene that's not carried. So both the father and the mother in this case are very tall. Okay, so this is tall because this is dominant, that's tall, but no, they don't have a 50-50 chance. All their kids are going to be tall. Sorry, just kidding. All their kids are going to be tall. However, their kids are going to be carrying this gene, and if they were to marry somebody that now this is the offspring here. So here's the offspring. This is what they had, and they married someone short. Now the second generation, this is second generation, this would be the first generation. Second generation is going to have some 50% chance of being short. So there's, my brain was jumping ahead of myself. I'm sorry about that. So these are all tall with two tall parents, but because of this one recessive gene, this is passed on to, and is still carried in their kids, and then their grandkids, if they were to marry someone short, definitely could have some short people come out of for their offspring. 50% chance, which is pretty big. All right, what do the letters on the axis of the Punnett square represent? Well, these are going to be what I was showing you up here, the father and the mom, so, or the genes, literally is the genotype. Okay, so genotype number 1 is always going to be on the top. So if we have, let's say this is, you know, the hair color gene, and then genotype number 2 is going to be on the side. So maybe it's little a. And so now we go through and make our Punnett square, and we have, you know, maybe this is brown hair, and this was so this would be the phenotype. And then the phenotype over here, this is blonde. So now we have a 50% chance of getting brown hair and a 50% chance of getting the blonde hair. Alright, so monohybrid cross is just going to be 4 squares. The ratio is always going to be just 2, so it's going to be how many dominant versus how many are recessive. Okay, and it's only for one trait. Okay, so I was just testing hair color. I was just testing height. Okay, for one trait. That's very, 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 very important. Okay, whereas a dihybrid cross is going to be a little more complex. Okay, so a dihybrid cross is going to have 16 squares, and its ratio is always going to be dominant both the first one and the second one are dominant. The first one is dominant, the second one is recessive. The first one is recessive, the second one is dominant, and then the last number is always going to be, they're both recessive. Recessive, recessive. Okay, so we get something like 9, 3, 3, 1, where this one would be something like 3 to 1. So the ratios are much different, and die means 2, so we're testing for 2 traits. And these are usually traits that follow each other. So freckles and red hair, or eye color and hair sometimes are similar. So dihybrid is going to be two traits. All right, so it says, how did Mendel's dihybrid cross help him develop his second law? So his second law was independent assortment. Okay, and independent assortment means that you have the ability to get any gene that just depends on which ones are expressed and the combination of these genes. Okay, so down here I will do a dihybrid cross. Whoa. Let me make sure. Actually, I think let's go ahead and go down here and make a dihybrid cross. So if you want to do a dihybrid cross with me, okay, so this is always going to be for 2 genes. So let's do yellow and tall. So I'm going to do yellow peas. And let's do Y is going to be dominant for yellow. Okay, and little y is going to be green. And then let's also do round or wrinkled.
Ulysses Casillas, report to the office, please. Ulysses Casillas, report to the office.
And little r will be wrinkled.
Office, please.
Okay, so we have big Y, little r, times little y, big R, little r. Okay, so we have one that is yellow and wrinkled, and one that is green and round, and we're going to put those together. So the very first thing we have to do is we have to do color first, and then we have to do peas shape second, and you do 2 monohybrid crosses first. Okay, so the first step, monohybrid cross for color, we're going to have big Y, little y, right here. We're going to just bring that right down here. And then the second one is going to go on the side, little y, little y. So we're going to have big Y, little y, little y, little y, big Y, little y, little y, little y. Okay, and then we do the shape. And we're going to do little r times big R, little r. Big R, little r, big R, little r, little r, little r, little r, little r. Okay, so now you're going to take what's inside of these monohybrid crosses and you're going to make it the dihybrid cross. Okay, so we'll just draw it here. Okay, so this first one is going to go right up on top. Big Y, little y. We have
Ashton Wilson, come to the office, please. Ashton Wilson, come to the office.
big Y, little y. So 2 big Y, little y's, 2 little y, little y's. We have big R, little r, big R, little r, little r, little r, little r, little r. Okay, so this one's going to be big Y, big R, little r. Little y, big R, little r, little y, big R, little r. Big Y, big R, little y. fill this in. Doesn't matter how you do it. What order, I mean. Okay, so now you have to figure out how many you have of each. So, do we have any big Y, big Y, big R, big R? No. Do we have any big Y, big Y, big R, little r's? No. Okay, so big Y, big R, little r. We have 1, 2, 3, 4. Okay, and then we have little y, I guess big Y, little r. We have none. So those are the ones that would be that color. Then we have the next ones that are going to be little y's, with a big R, which we have none of those. Big Y, little r, and big R together. So we have 1, 2, 3, 4 of those. And then we had, 4 of these. We'll put those over here. And then our last one that we have any of them are these. We have 4 of these. So now we have to transfer that to our phenotype. So our phenotype is, we can do yellow and round, which would be dominant, which is our first number. We could do yellow and wrinkled. We could do green and round. Or we can do green and wrinkled. So yellow and round, we have 4. Yellow and wrinkled, we have 4. Green and round, we have 4. Green and wrinkled, we have 4. So our ratio for a dihybrid cross is 4, 4, 4, 4. Okay, so let's go back up here. Alright, so now we have our 4 exceptions to Mendel's 2 laws, which is the independent assortment and this law of segregation. So incomplete dominance, that is going to be our first one. So this is number 1. And this is going to be when something blends. Okay, so we had the 4 o'clock flowers, where we crossed the red and the white, and instead of getting whatever one was dominant and recessive, we got pink. Also the betta fish, they do this, and we read about that in the book, and so this is the blending concept. Okay, so codominance is our second one, and codominance is different because instead of being blended, they are both expressed in the same way, so these are both expressed. And so is that in codominance. They're both expressed. However, they are different because the codominance usually has them separate. So this was the example of the black and white chicken making the black and white spotted chicken. Okay. Codominance also has a lot to do with blood typing. Okay, and so can 2 people with B blood have a child with O blood? Yes, that is something that can happen. So when we're looking at codominance here, codominance means that both can be expressed. And so in blood typing, somebody with B or somebody with A blood A is going to, when you do a Punnett square, is going to be capital I with a little a, and then a little I. That would be its heterozygous. B is going to be capital B little I, and then O is going to be two little i's, and then obviously AB is going to be capital I, big A, capital I, B. Okay. So if you were making a Punnett square, two people with B, they're going to have big I, B with a little I, big I, B with a little I. So we'd have big I here with a B. We'd have big I, B with a little I, big I, B with a little I, and then our last one would be O blood, because that's O. So you can have two parents that have type O blood because they're both going to be, can be dominant. So when that happens, or they can both be recessive. And if they're both going to be recessive, you're going to get O blood. This also could be somebody with A blood. Two people with A blood can have a baby with O blood. Okay, in fact, I'm A blood, and Elena is O blood. So that is how that happens. Okay, our third type is called a polygenetic trait. This is the example that we used was eyes, and the book talks about eyes. And polygenetic trait is going to be multiple genes or multiple alleles that are all expressed as dominant and recessive. So you can have two dominant genes, so you can have like on your eye, you can have like the outer line be blue, oops, what the, blue, and the inner area here be more of a green color. Okay, and so we can have two things that are dominant here, and or you could have a brown eye that had like some green in the middle here, and you get some of this blending where you see all of the different colors, and it's not necessarily just two, so it's a little different than what codominance is. Codominance doesn't have dominant and recessive either. You just kind of have two genes that are both expressed. This is more than just two genes. This is a lot of genes being expressed, and they can either be dominant or recessive, and they can blend together. Okay. And then the third type, or our fourth one, is going to be, number 4 is going to be epistasis. And epistasis is the epi means to block, and so it's a gene that can that blocks other genes. And a big example of this is the albino. So the albino gene goes in and it blocks all the rest of the genes from producing any color. So that's kind of an interesting genetic anomaly. All right, and then this one talks about the environment, okay, affecting your phenotype. Okay, so we talked about skin color, you know, being in the sun more is going to change it. You know, your genes might say you're supposed to be skinny, but you eat a really high fatty diet and you might not be skinny. So your parents might be skinny and you live a different lifestyle than them. Or vice versa, you know, maybe genetically you're supposed to be a bigger person, but you're really fit and you work out a lot and you eat a really healthy diet, then you're going to not be what your genetics say you're supposed to be. Same with, if you're supposed to have healthy lungs and heart, but you choose to smoke, environment can affect the phenotype, and it can change what your genes are supposed to be. All right, so I actually answered this one right up here with my Punnett square right there, because it says answer with a Punnett square. But this one says type A and type B blood. So type A, the only thing that would be different is if one of these this one was an A, so then we'd have one person with AB, or would have one chance of AB blood. So I'll draw out that Punnett square. So if you had I, capital A, I, capital B, little I, you're going to have one person with AB blood. This person is going to have B blood. This one is going to have A blood. And this one is going to have O. So when parents have, one parent has A blood and one parent has B blood, the possibilities could be A blood, B blood, AB blood, or O blood. So that's really interesting. So that's like the Punnett square. We couldn't just write 3 to 1 there. It would actually have a 1 to 1 to 1 to 1 probability. So kind of interesting. And then just make sure on your test there is one that kind of talks about the second generation. So first generation, you know, you take this and then you take one of these and move it to the next generation. So just do another Punnett square of the next generation, and that's what we would be talking about there. Doing what could happen in the next generation if somebody married somebody that was short. All right, I hope this helped, and